So, we will continue with another speaker who is here with us physically, an associate professor from the University of Debrecen. This is a lovely city at the northeastern part of Hungary, where he serves in the Department of Agricultural Chemistry and Soil Sciences. He holds a CSc and habilitation. Throughout his career, our speaker has demonstrated expertise in environmental awareness, plant nutrition, physics, agricultural chemistry, and analytical chemistry. Notably, he and his research group have designed a soil microbiology experiment to be conducted aboard the MayaSat-1 biological incubator as part of the aforementioned Mission Possible 2025. Ladies and gentlemen, please welcome Dr. Imre Vágó from Hungary.
Thank you very much. To tell the truth, as Zoltán said already, I am from Debrecen, which is a really eastern part. Thank you. Eastern part of Hungary, about 30 kilometers from the Romanian border and 100 kilometers from the Ukrainian border. Now we have the population is about 200,000. Of the city, but we have a big university, the biggest university at that time in Hungary, and we have more than 30,000 students and 2,000 colleagues. It means that it's about 15% of the population of Debrecen, and it can be also really feel at the city too. Our university has 13 faculties, really different one, from agriculture to medicine and music and so on. Sometimes we say that at our university you can study almost everything from geology to theology. One of the faculties is our faculty. This is the Faculty of Agriculture and Food Science and Environmental Management. And we have 20 institutes and departments in our faculty. And I am from the Institute of Agricultural Chemistry and Soil Science, as it was already mentioned by Zoltán. Our institute has two working groups. Okay, you can imagine one is the agricultural chemistry group, the other soil science group. The lectures will be held separately, the two groups, but the research work, we are collaborating on several frames, several projects. To tell the truth, we have, let's say, broad international connections to the strongest and more intensive or most intensive connection. We have Germany, but we have also some connection to the George Washington University in Washington, D.C., in Japan, Indonesia, and former we had also contact to Ukrainians. It is terminated at the last time, sorry. Okay. Now about for us almost new products. This is the plant production in the space. Okay, and some words about the history. But I mean it is the people here are or the colleagues here are. Really informed about it, but I want to mention this that the first step was in 1946. At that time, the NASA V2 rockets were starting a spaceship with plant materials into the and they want to see the effect of the cosmic radiation. The samples were not recovered at that time. It was not possible technically, but later on, corn kernels launched shortly also, and it was followed by rye and cotton. Oh, okay. I have to say, I feel it that I am not really good at the time in English, despite I was learning it more than 50 years, half a century. Okay. Later on in 1966, the space flights of 2 ducks received more press coverage, but there were also vegetable seeds, lettuce, beans, cabbage on board. The yield of these seeds ripened on the Earth's back. They were bigger than the parallel which was not launched in the space. The reason was not known at that time. It was mentioned as perhaps the cosmic radiation has a positive effect, but because of the small amount of samples, it cannot be proven. The next step was in 1971. The astronauts of spaceship Apollo 14 carried seeds of wood on their lunar mission. The seeds rotated altogether 34 times around the moon. After returning the space, from the space, the seedlings was germinated, and hundreds of the small plants were distributed. At that time, about 50 trees we have in the different corner of the Earth with this moon traveling plants. Now, let's see, the crop production is already in operate. It is going on. The first chamber was for this purpose the vegetation production system is let's see, a space garden at the space station. The veggie is designed for help of NASA study plant, but it produces also some food for the astronauts and improve the diets and the well-being of the astronauts. This veggie, let's say this is a small, because it is just like a suitcase, and typically can grow 6 plants in this veggie. Each plant grows in, let's say, pillow or production pot, small production pot, filled with clay-based nutrient medium and fertilizer. The pillow help to distribute water, nutrients, and air in a healthy balance around the roots. Otherwise, the roots would either drown or dry out as the liquids of the would tend to disperse of droplets. In the absence of constant gravity, plants use other environmental factors such as light to orient and guide the growth. Light-emitting diodes positioned above the plant produce a spectrum of light that is appropriate for the plant's growth. It is designed for the plant's growth. Because the plants reflect a lot of green light and use more red and blue wavelength, the vegetable chamber is typically magenta pink. Still in operation today, the veggie, it has successfully grown different plants, different crops, including the types, three types of lettuce, Chinese kale, a kind of mustard, red Russian kale, and even zinnia flowers. What we can see here, this is an astronaut can harvest the plants from the veggie. This is lettuce, and they said, the astronauts said that with balsamic vineyard, the taste was really good, and they were enjoying the taste. Now, the other equipment which was designed for the production of crop in the space. This is the Advanced Plant Habitat, APH is the abbreviation. It's a plant growing chamber similar to the veggie, but it has a larger side and fully enclosed and automated. The shoots area, this is 700 square centimeter. The roots area, 1800 square centimeter. The shoot height is 43 centimeter, and the root height is 5 centimeter. The equipment has 180 sensors which monitors the processes. Moisture and nutrient supply is automatic controlled. Sensor provide autonomous function based or programmed experimental design. Includes an active thermoregulation. It means that with the cooling and heating system, the temperature can be hold between the 18 and 30 Celsius. Controls grows also the carbon dioxide level, light intensity and spectrum, humidity, and it can work in one step for 135 days. It means it's really long, and it is enough for plants which have a short vegetation period. In 2018, short-duration dwarf wheat was also successfully grown in that APH system alongside the fieldgrass. Research is focusing on genetic changes resulting from weightlessness. It is well known that the bones and muscles of the astronauts began to atrophy in the persistent absence of gravity now, and we have to calculate that the similar phenomenon can be also find in the case of the plants. The lignin production will be probably also minimized. And ongoing research is being carried out into the effect of the persistent absence of the gravity of plants to see whether they can produce normal long-term vegetative development, a state of weightlessness. And you can see here the draft wheat which was grown in the space in the chamber, what I was mentioned. After 5 weeks, we can see the plants here. It means that it is successful. It was successful. Now about the role of the University of Debrecen in space crop production. To tell the truth, we was joining shortly before to the Honor program, and in frame this Honor program, the Debrecen University, the research of the Debrecen University are also involved. The first step was that the Department of Applied Plant Biologist at our university were working on the breeding of space pepper, capsicum, or in German and Hungarian, this is the paprika. The aim to produce edible pepper that can be grown in soil-free hydroponic condition in confined spaces under artificial light. It is practically advantageous if the flowers develop simultaneously. Pollination is by self-fertilization, and resulting the crop developed rapidly. The aim is that not only the fruit but also the plant leaves themselves should be edible. The taste of the leaves of pepper, this pepper variety, the Hungarian Enigma sweet, is really similar to the Argula, according to the most organoleptic analyzer. It means that it was successful. And you can see here the terrestrial version of the pepper growing chamber, which is designed for the space travel. And here are the paprika, which was produced in this chamber under the fully artificial light, and all of the factors was artificial. Now, to tell the truth, the biggest problem of space plant production, there is no soil on the surface of the planets. We know. And we know that it is possible to grow crops without soil, but not on a scale that would provide sufficient food for the astronauts. Growing plants in soil is much better solution, we know it, as it binds moisture and provides it water to the plants at the constant time. However, the soil take a lot of volume, and because of the big mass, the required amount cannot be transported into the space or to another planet. On the International Space Station, the necessary food supplies are available, but for the multi-year journey or base on the other planets, that is, of course, not a good solution. It means that we should also grow plants. What is the possible solution? Sorry? Okay. If we want to set up a basis on the other planets, we need to provide food suppliers on the ground. The simplest way to do this is to the weathered rock of the planets. Okay, we have to find microorganisms, and these are present in the Earth's soils, and they are able to contribute to the slow weathering of the soil-forming rocks. But exploring and transforming the nutrients they contain, they will be made available, the nutrients for the plants. They also carry out the transformation of human wastes so that they can also supply organic matter to the rock soil system. Okay. In our current collaborative research, we are investigating how microbial communities in soil samples from nature conditions in Hungary change in response to orbital radiation, which is, of course, which can also influence the living beings. After sampling of the soil, we determined the phospholipid fatty acid PLFA patterns in the soil, one of the most commonly used methods to investigate microbial community structures. Phospholipid fatty acid are found in cell membranes of living organisms, and these are different. It has different contribution in the different living beings. It means the cell membranes of fungi are typically composed of different PLFAs, than that of bacteria. The PLF spectra of microorganisms present on soil in soil are identifying using gas chromatography mass spectrometer coupled analysis system, and after a proper calibration the quantity of the different components can be also calculated. Now, and what we done and what we want to do at this field, we were taking already soil samples from the natural conditions of Hungary and from Ecuador, okay. We were determining the composition of the microbial community from the sample. Sample is stored under conditions similar to those used during the MAIASat-1 mission. Send the samples to the MAIASat mission. Before the sending, we will again control the microbiological properties, and the controls will be held also on the earth and on the ground, similar that it is under MAIASat-1, and we will heat it for a short time up to 40 centigrade for simulation of the returns of the MAIASat. After sampling, after sample re-enter, the phospholipid fatty acid pattern of the samples, and from this microbial community structure will be also determined, and after the result we hope we can get the answer on the question which group of the microorganisms are most resistant for the space travel. Okay, the microorganism composition of the sample, which is already prepared for the further work, you can see here. It means that is okay. It is not a cheap method, but it is really good method, and how does it say? Really precise also. This was what I said about the microbiological composition. Okay. And our objective for the future is developing a vacina, bacteria and fungi combination that can tolerate the effect of the space travel. They have the ability to extract nutrients from the residues of the soil-forming parent materials. It is able to degrade the transforming organic matter, and this will provide the basis for some of the nutrient replacement based on crop production on the space basis. To achieve this, we still need a lot of experiments, or even countless experiments, and in which we will be able to simulate the space conditions. And thank you very much for your kind attention.
Thank you very much, Professor. Do you have any questions, ladies and gentlemen, about this topic to the professor from Debrecen? was great to hear the mentioning of paprika. It's
Yeah.
very important in the Hungarian cuisine. No more questions. Thank you very much. Thank you.
Thank you.
Thank you, Professor.
Thank you.